स्टार्ट करने जा रहे थर्ड यूनिट सेल स्ट्रक्चर एंड फंक्शंस. Biology is the study of living organisms. The detailed description of their form and appearance only brought out their diversity. It is the cell theory that emphasized, emphasized the unity underlining this diversity of forms, that is the cellular organization of all the life forms. A description of cell structure and cell growth by division is given in the chapters comprising this unit. Cell theory also created a sense of mystery around living phenomena, that is physiological and behavioral processes. This mystery was the requirements of integrity of cellular organization for living understanding the physiological and behavioral processes. One can take a physicochemistry approach and use cell-free systems to investigate. This approach enables us to describe the various processes in molecular terms. The approach is established by analysis of living tissues for elements and compounds. It still it will tell us what two types of organic compounds are present in living organisms. In the next stage, one can ask the question, what are these compounds along inside a cell, and in what way they carry out gross physiological processes like digestion, excretion, memory, defense, recognition, etc. In other words, we answer the question, what is the molecular basis of all physiological processes. It can also explain the abnormal behavior processes that occur during any disease conditions. This physicochemical approach to study and concepts and techniques of physics and chemistry are of concepts and techniques of physics and chemistry are applied to understand biology. In chapter 9 of this unit, a brief description of biomolecules is approved. G. N. Ramachandran, 1922 to 2001, an outstanding figure in the field of protein structure, was the founder of Madras School of conformational analysis of biopolymers. His discovery of the triple helical structure of collagen, published in Nature in 1954, and his analysis of the allowed conformations of proteins through the use of Ramachandran plot rank among the most outstanding contributions in structural biology. He was born on 8 October 1922 in a small town not far from Cochin on the southwestern coast of India. His father was a professor of mathematics at a local college and thus had considerable influence in shaping Ramchandran's interest in mathematics. After completing his school years, Ramchandran graduated in 1942 at the top University of Madras. He received a PhD from Cambridge University in 1949. While at Cambridge, Ramachandran made models of the alpha helix and beta sheet structures that directed his attention to solving the structure of collagen. He passed away at the age of 78 on April 7, 2001. Chapter 8, Cell, the Unit of Life. When you look around, you see both living and non living things. You must have wondered and asked yourself, what is it that makes an organism living? Or what is it that an inanimate thing does not have, which a living thing has? The answer to this is the presence of the basic unit of life, the cell in all living organisms. All organisms are composed of cells. Some are composed of a single cell and are called unicellular organisms, while others, like us, composed of many cells, are called multicellular organisms. What is a cell? Unicellular organisms are capable of independent existence and performing the essential functions of life. Anything less than a complete structure of all of cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and its improvement leading to the electron microscope revealed all the structural details of the cell. Cell theory. In 1838, Matthias Skeldon, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells, which form tissues of the plant. At about the same time, Theodore Schwann, in 1839, a British zoologist, studied different types of animal cells and reported that cells had a thin outer layer which is today known as the plasma membrane. He also concluded, based on his studies on plant tissues, that the presence of cell wall is a unique character of the plant cells. On the basis of this, Chavan proposed the hypothesis that the bodies of animals and plants are composed of cells and products of cells. Chavan, Skeldon and Chavan together formulated the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow, 1855, first explained that cells divided and new cells are formed from pre-existing cells. Omnis cellula e cellula. He modified the hypothesis of Skeldon and Schwann to give the cell theory a final shape. Cell theory, as understood today, is all living organisms are composed of cells and products of cells. All cells arise from pre-existing cells. An overview of cell. You have earlier observed cells in an onion peel or a human cheek. Cells under the microscope. Let us recollect their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human cheek have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane bound structure called a nucleus. This nucleus contains the chromosomes, which in turn contain genetic material. DNA cells that have membrane bound nuclei are called eukaryotic, whereas cells that lack the membrane bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cells, a semi-fluid matrix called cytoplasma occupies the volume of the cell. The cytoplasma is the main arena of cellular activities in both the plant and animal cells. Various chemical reactions occur in it to keep the cells in the living state. Besides the nucleus, the eukaryotic cells have other membrane bound distinct structures called organelles, like the endoplasmic reticulum and Golgi complex, lysosomes, mitochondria, microbodies, and vacuoles. The prokaryotic cells lack such membrane bound organelles. Ribosomes are non membrane bound organelles found in all cells, both eukaryotic as well as prokaryotic within the cell. Ribosomes are found not only in the cytoplasma but also within the two organelles, chloroplasts in plants and mitochondria, and on rough endoplasmic reticulum. Animal cells contain another non-membrane bound organelle called centrosome, which helps in cell division. Cells differ greatly in size, shape, and activities. Figure 8.1. For example, mycoplasma, the smallest cells are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. The largest isolated single cell is the egg of an ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Nerve cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboid, thread-like, or even irregular. The shape of the cell may vary with the function they perform. Prokaryotic cells. The prokaryotic cells are presented by bacteria, blue-green algae, mycoplasma, and PPLO, pleuropneumonia-like organisms. And they are generally smaller and multiply more rapidly than the eukaryotic cells. Figure 8.2. And they may vary greatly in shape and size. The four basic shapes of bacteria are bacillus, rod-like, coccus, spherical, vibrio, comma-like, spirillum, spiral-like. The organization of the prokaryotic cells is fundamentally similar, even though prokaryotes exhibit a wide variety of shapes and functions. All prokaryotes have a cell wall surrounding the cell membrane, except in mycoplasma. The matrix fluid matrix filling the cells in the cytoplasma. There no well defined nucleus. The genetic material basically naked, not enveloped by a nucleus membrane. In addition to the genomic DNA, the single chromosome circular DNA, many bacteria have small circular DNA outside the genomic DNA. These smaller DNA are called plasmids. The plasmid DNA confers certain unique phenotypic characters to such bacteria. One such character is resistance to antibiotics. In higher classes, you will learn that this plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelles like the one like the ones in eukaryotes are found in prokaryotic cells, except for ribosomes. Prokaryotes have something unique in the form of inclusions, a specialized differentiated form of cell membrane called a mesosome. is the characteristic of prokaryotes. They are essentially enfolding of cell membrane, cell envelope, and its modification. Most prokaryotic cells, particularly the bacterial cells, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure, that is the outermost glycocalyx, followed by the cell wall, and then the plasma membrane. Although each layer of the envelope performs distinct functions, they act together as a single protective unit. Bacteria can be classified into two groups on the basis of differences in the cell envelopes and the manner in which they respond to the staining procedure developed by Graham Wise. Those that take the take up the gram stain and the gram positive, and the others that do not are called gram negative gram negative bacteria. Glycocalyx differs in composition and thickness among different bacteria. It could be a loose sheath called the slime layer in some, while in others it may be thick and tough called the capsule. The cell wall in others, the cell wall determines the shape of a cell and provides the strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structurally to that of the eukaryotes. A special membranous structure is the mesosome, which is formed by the extensions of plasma membranes into the cell. These extensions are in the form of vesicles, tubules, and lamellae. They help in the cell wall DNA replication, and distribution to daughter cells. They also help in the respiration secretion processes to increase the surface area of the plasma membrane and enzymatic content. In some prokaryotes like cyanobacteria, there are other membranous extensions into the cytoplasm called chromatophores, which contain pigments. Bacterial cells may be motile or non-motile. It motile, they have thin filamentous extensions from their cell wall called flagella. Bacteria show a range in the number and arrangements of flagella. Bacterial flagellum is composed of three parts: filament, hook, and basal body. The filament is the longest portion of extent from the cell surface to the outside. Besides flagella, pili and fimbriae are also surface structures of the bacteria but do not play a role in motility. The pool are elongated tubular structures made of protein social protein. The fimbriae are small bristle-like fibers sprouting out of the cell. In some bacteria, they are known to help attach the bacteria to rocks in streams and also to the host tissues. Ribosomes and inclusion bodies. The prokaryotes ribosomes are associated with the plasma membrane of the cell. They are about 15 nanometer by 20 nanometer in size and are made of two subunits, 50 years and 30 years unit, which when present together from 70 years prokaryotic ribosomes. Ribosomes are the site of protein synthesis. Several ribosomes may attach to a single mRNA and form a chain called polyribosomes. Polysomes. The ribosomes of a polysome translate the mRNA into proteins. Inclusion bodies. Reserve material in prokaryotic cells are stored in the cytoplasm in the form of inclusion bodies. And these are not bound by granules, cyanophysin granules, and glycogen granules. Gas vacuoles are found in blue, green, and purple and green photosynthetic bacteria. Eukaryotic cells. Eukaryotes includes all the proteins, plants, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization and cytoplasma through the presence of membrane-bound organelles. Eukaryotic cells possess an organized nucleus with the nuclear envelopes. In addition, eukaryotic cells have the variety of complex locomotory and cytoskeleton structures. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the former possess cell walls, plastids, and the large central vacuole which are present in the animal cells. On the other hand, animal cells have centrioles which are absent in almost all plant cells. Figure 8.3. 8.3 diagram showing plant cell and animal cell. Let us now look at the individual cell organelles to understand their structure and functions. Cell membrane. The detailed structure of the membrane was studied only after the advent of the electron microscope in 1950s. Meanwhile, chemical studies on chemical studies on the cell membrane, especially in human red blood cells (RBCs), enabled the scientists to deduce the possible structure of plasma membrane. These studies showed that the cell membrane is mainly composed of lipids and proteins. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer sides and the hydrophobic tails towards polar head towards the outer side and the hydrophobic tails towards the inner part. This ensures that the nonpolar tail of saturated hydrocarbons is protected from the aqueous environment, which is located in figure 8.4. In addition to phospholipids, membrane also contains cholesterol. Later biochemical investigation clearly revealed that the cell membrane also possesses proteins and carbohydrates. The ratio of protein and lipid varies considerably in different cell types. In human beings, the membrane of the erythrocytes has approximately 52% protein 52% protein and 40% lipids. Depending on the age of extraction, membrane proteins can be classified as integral, and peripheral proteins lie on the surface of the membrane, while the internal integral proteins are partially or totally buried in the membrane. An improved model of the structure of cell membrane was proposed by Singer and Nicholson in 1972, widely accepted as fluid mosaic model, which is located in figure 8.4. According to this, the fluid structure Quasi fluid nature of lipid enable lateral movement of proteins within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of functions like cell growth, formation of intercellular functions, secretion, and endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane above without any requirement of energy, and this is called the passive transport. Neutral solutes may move across the membrane by the process of simple diffusion along the concentration gradient, that is from higher concentration to lower concentration. Water may also move water by diffusion is also osmosis, as the popular as the polar molecules and cannot pass through the non-polar lipid bilayer. This require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions and molecules are transported across the membrane against their concentration gradient, that is from lower to the higher concentration. Such a transport is an energy dependent process in which ATP is utilized, and it's called active transport. Like for example, the sodium ion potassium ion pump. Cell wall. As you may recall, now non-living rigid structures called the cell wall from an outer covering for the plasma membrane of fungi and plants. Cell wall not only gives shape of to the cell and protects the cell from mechanical damage and infection. It also helps in cell to cell interaction and provides barrier to understand undesirable macromolecules. Algae have cell wall made of cellulose, galactans, mannans, and minerals like calcium carbonate, while in other plants it consists of cellulose, hemicellulose, pectins, and proteins. The cell wall of the young plant cells. The primary wall is capable of growth, formed on the inner towards membrane side of the cell. The middle lamella is the layer main of calcium pectate, which holds or glues the different neighboring cells together. The cell wall and middle lamellae may be traversed by plasmodesmata, which connect the cytoplasma of neighboring cells. Endomembrane system. While each of the membranes organs is distinct in terms of its structure and function, many of these are considered together as an endomembrane system because their functions of are coordinated. The endomembrane system include endoplasmic reticulum, ER, Golgi complex, lysosomes, and vacuoles. Since the functions of the mitochondria, chloroplast, and peroxisomes are not coordinated with the above components, these are not considered as part of the endomembrane system. The endoplasmic reticulum. Electron microscopic studies of eukaryotic cells reveal the presence of the network or reticulum of tiny tubular structures scattered in the cytoplasm. That is called the endoplasmic reticulum, which is located in figure 8.5. Hence, endoplasmic reticulum divides this intracellular space into two distinct compartments, that is luminal inside endoplasmic reticulum and extraluminal cytoplasma compartments. The endoplasmic reticulum often shows ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosomes on their surface is called rough endoplasmic reticulum RER. In the absence of ribosomes, they are they are they appear smooth and are called smooth endoplasmic reticulum SER. RER is frequently observed the cell actively involved in protein synthesis and secretion. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum of the endoplasmic reticulum often shows ribosomes attached to their outer surface. The endoplasmic reticulum bearing ribosomes on their surface is called rough endoplasmic reticulum RER. In the absence of ribosomes, they appear smooth and are called smooth endoplasmic reticulum. RER is frequently observed in cells actively involved in protein synthesis and secretion. They are extensive and contributions continuous with the outer membranes of the nucleus. The smooth endoplasmic reticulum of the major site for synthesis of lipid. In animal cells, lipid-like steroidal hormones are synthesized in SER, smooth endoplasmic reticulum. Golgi apparatus. Camillo Golgi in 1898 first observed densely stained reticular structures near the nucleus. These were later named Golgi bodies after him. They consist of many flat, disc-shaped sacs or cisternae of cisternae of 0.5 micrometer to 1 micrometer diameter. Figure 8.6. Golgi apparatus. These are stacked parallel to each other. Varied number of cisternae are present in the Golgi complex. The Golgi cisternae are concentrically arranged near the nucleus or with distinct convex cis or the forming face and concave trans or the manufacturing phase. The cis and the trans phases of the organelle are entirely different, interconnected. The Golgi apparatus principally performs the functions of packaging materials to be delivered either to the intracellular structure targets or secreted outside the cell. Materials to be packed in the form of vesicles from the AR fuse with the cis phase of the Golgi apparatus and move towards the manufacturing phase. This explains why the Golgi apparatus remains in close association with the endoplasmic reticulum. The number of proteins synthesized by ribosomes on the endoplasmic reticulum are modified in the cisternae of the Golgi apparatus before they are released from its trans phase. The Golgi apparatus is the important site of formation of glycoproteins and glycolipids, lysosomes. These are membrane-bound vesicular structures formed by the process of packaging in the Golgi apparatus. The isolated lysosomal vesicles have been found to be very rich in almost all types of hydrolytic enzymes, hydrolysis, lipases, proteases, carbohydrates, optimally active in the active at the acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acids. Vacuoles. The vacuole is the membrane bound space found in the cytoplasm. It contains water sap to excretory the plants. The chronoplast facilitates the transport of a number of ions and other materials against concentration gradients into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, this conus on the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protistas, food vacuoles are formed by engulfing the food particles. Mitochondria, sign mitochondrion, unless specifically stained are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological activity of the cells in terms of shape and size also. Considerable degree of variability is observed. Typically, it is sausage shaped or cylindrical, having a diameter of 0.2 to 1 micrometer, average 0.5 micrometer, and the length 1 to 4.1 micrometer. Each mitochondrion is a double membrane bound structure with the outer membrane of the inner membrane dividing its lumen distinctly into two aqueous compartments, that is the outer compartment and the inner compartment. The inner compartment is filled with the dense homogeneous substance called the matrix. The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms the number of folding enfolding called the cristae towards the matrix. Figure 8.7. The cristae increase the surface area. The two membranes have their own specific enzymes associated with the mitochondrial function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called powerhouses of the cell. The matrix also possesses single cellular DNA molecule, a few RNA molecules, ribosomes 70s, and the components are required for the synthesis of proteins. The mitochondria divide by fission. Plastids. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscopes. They are large, bear some specific pigments, thus imparting specific colors to the plants. Based on the type of pigments, plastids can be classified into chloroplasts, chromoplasts, and lycoplasts. The chloroplasts contain chlorophyll and the carotenoids pigments which are responsible for trapping light energy essential for photosynthesis. This in the chromoplast, fat soluble carotenoids pigments like carotene xanthophyll and other are present. This gives the part of plant a yellow, orange, and red color. The leucoplasts are the colorless plastids of various shape and size with stored nutrients. Amyloplasts store carbohydrates, starch, example potato. Elenoplasts store oils and fats, whereas the aleuroplasts store proteins. Majority of the chloroplasts of the green plants are found in the mesophyll cells on the leaves. These are lens-shaped, oval, spherical, discoid, or even ribbon-like organelles having variable length 5 to 10 micrometer and width 2 to 4 micrometer. Their number varies from 1 per cell of the Chlamydomonas. The green algae of 20 to 40% in the mesophyll, like mitochondria. The chloroplasts are also double membrane bound. Of the two, the inner chloroplast membrane is relatively less permeable, and the space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattened membranous sacs called the thylakoids are present in the stroma, figure 8.8. Thylakoids are arranged in stacks like the piles of coins called grana, singular granum, or the intergranal thylakoids. In addition, there are of different grana. The membrane of the thylakoids encloses space called the lumen. The stroma of the chloroplast contains enzymes required for the stranded circular DNA molecules and ribosomes. Chlorophyll pigments are present in the thylakoids. The ribosomes of the chloroplasts are smaller, 70s, than the cytoplasmic ribosomes, 80s. Ribosomes. Ribosomes are the granular structures first observed in under the electron microscope as dense particles by George Palladay in 1953. They are composed of ribonucleic acid (RNA) and proteins and are not surrounded by any membrane. The eukaryotic ribosomes are 80s while the prokaryotic ribosomes are 70s. Each ribosome has two subunits, larger and smaller subunits. Figure 8.9. The two subunits of 80s ribosomes and 60s and 40s, while that of 70s ribosomes are 50s and 30s. Here, yes, Swedberg's unit stands for the dimension coefficient. It is indirectly measure density and size. Both 70s and 80s ribosomes are composed of two subunits. Cytoskeleton, the elaborate network of filaments, its proteinous structures consisting of microtubules microfilaments, and intermediate filaments present in the cytoplasm of it's collectively is referred to as the cytoskeleton. The cytoskeleton in a cell are involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Cilia and flagellum are here like outgrowths of the cell membrane. Cilia are small structures which work like oars, causing the movement of either the cell. or the surrounding fluid. Flagella are comparatively longer and responsible for cell movement. The prokaryotic bacteria also possess cell flagella, but these are structurally different from that of the eukaryotic flagella. The electron microscopic study of the cilium or the flagellum show that they are covered with plasma membrane. Their core, called the axoneme, axoneme possesses a number of microtubules running parallel to the long axis. The axoneme usually has nine doublets of radially arranged peripheral microtubules and a pair of centrally located microtubules. Such an arrangement of axonal microtubules is referred to as the 9 plus 2 array, figure 8.10. The central tubules are connected by bridges and is also enclosed by a central sheath, which is connected to one of the tubules of each peripheral doublets by a radial spoke. Thus, there are nine radial spokes on the peripheral doublets are also inter- interconnected by links. Both both the cilium and flagellum emerge from central-like structures called the basal bodies. Centrosome and centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles in centrosomes lie perpendicular to each other. Each has an organization like the cartwheel. They are made up of nine evenly spaced peripheral fibrils of tubulin proteins. Each of the peripheral each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous, are called the hub, which is connected with tubules of the peripheral triplets by radial spokes made of protein. The centrioles form the basal body of the cilia or flagella, the spindle fibers that give rise to spindle apparatus during cell division in animal cells. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. The interface nucleus of the cell, when it is not dividing, has highly extended and elaborate nucleoprotein fibers called chromatin. Nuclear matrix and one or more spherical bodies called nucleolus, figure 8.11. New electron microscopy has revealed that the nuclear envelopes, which consists of two parallel membranes with the space between 10 to 15 nanometer, called the perinuclear space, forms a protein barrier between the materials present inside the nucleus present inside the nucleus, and membrane usually remains. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it at a number of places. The nuclear envelope is interrupted by minute pores which are formed by the fusion of its two membranes. These nuclear pores are the passages through which movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. Normally, there is only one nucleus per cell. Variations in the number of nuclei are there are have more than one nucleus per cell. Some mature cells even lack nucleus. Example: erythrocytes of many mammals and sieve tube cells of vascular plants. You would consider these cells as living. The nuclear matrix or the nucleoplasma contains nucleolus and chromatin. Nucleoli are spherical structures present in the nucleoplasma. The content of nucleolus is continuous with the rest of the nucleoplasma as it's as it is not a membrane bound structure. It is a site for active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. You may recall that the interface nucleus have a loose and the indistinct network of nucleoprotein fibers called the chromatin, but during different stages of cell division, cells shows a structured chromosomes in place of the nucleus. Chromatin contain DNA and some basic proteins called histones, some non-histone proteins, and also RNA. A single human cell has approximately 2-meter long thread of DNA distributed among its 46 23 pairs chromosomes. You will study the details of DNA packaging in the form of a chromosome in class 12. Every chromosome visible only in dividing cells essentially has a primary contribution or the centromere on the sides of which disc-shaped structures called kinetochores are present. Figure 8.12. Centromere holds two chromatids of a chromosome based on the position of the centromere. The chromosomes can be classified into four types. Figure 8.13. The metacentric chromosome has middle centromere forming two equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the middle of the chromosome, resulting into one shorter arm and one longer arm. And in case of acrocentric chromosome, the centromere is situated close to its and forming the centromere is situated close to its and forming one extremely short and one very long, whereas the telocentric chromosome has a terminal centromere. Microbodies. Many membrane bound minute vesicles called microbodies that contain various enzymes present in both plant and animal cells. तो यहाँ पर हमारा सेल द लिविंग ऑर्गेनिज्म सेल चैप्टर ख़त्म होता है और समरी स्टार्ट होती है। सेल द यूनिट ऑफ लाइफ, सेल द यूनिट ऑफ लाइफ चैप्टर यहाँ पर ख़त्म होता है। सो ये है उसकी समरी। सो ऑल द वेरी बेस्ट। थैंक यू सो मच फॉर वॉचिंग।